Okay, I guess we can start. Welcome back to Proteins and Protein Folding, now third edition. So today is going to be a little bit shorter. The main topic of today's workshop is going to be how does folding happen? And I also want to introduce to you another function that you can find in PyMol, which is the builder. So we can build our own little protein. And as well as I want to show you another software that you can use if you want to solve real life protein structure questions and help research if you want. Okay. So, but let's start off with a little review from last time. All right, so last time, if you remember, we talked a little bit about not only within a peptide, what kind of interactions there are, but also like looking at a bigger structure, how do these structures come to be, right? What we saw last time in PyMol, this whole big protein has a lot of different parts that have a defined structure. And for instance, we talked about the amino acid rotations, right? So because of the peptide bond, there's a certain limitation to how amino acids can rotate, and therefore what kind of conformation and what kind of structures they can build. Right? Just as a little reminder, we very shortly looked at what I actually don't know how to say it, a Ramachandran plot looks like, which basically shows us that there are three possible conformations that amino acids are usually in. Right? One of them was, for instance, the parallel beta-pleated sheet, an anti-parallel beta-pleated sheet, and a right-handed helix. Right? Most of other amino acids are unstructured. Right? After that, we looked a little bit more at bigger structures that these kind of secondary structures can form. For instance, we looked at the beta barrel. Last time in our protein, we also saw there were actually two beta barrels that we could observe. And also we talked a little about domains, and that domains, for instance, have different functions. Right? For instance, in this example, you can see like this is a transmembrane domain, right? So a part that is very hydrophobic and would sit in the membrane of a cell, right? So very often also, if proteins have similar domains and they share similar structures and domains, they also have similar functions that are associated with them. We'll go more into detail in the future about that topic. Okay. So let's start with something new. Well, kind of new. In the first workshop, I already told you a little bit, or I tried to actually ask you how proteins are made, right? Because today we want to look a little bit into how proteins get their structure, and that is somewhat connected to how the whole process of making a protein basically progresses in the cell, right? So if you remember, basically first what we have is DNA, right? Everybody knows DNA has all the information about our cells, and this is translated into RNA, right? Then leaves the nucleus and goes into the cytoplasm, where then a ribosome attaches itself and translates this mRNA sequence into an amino acid sequence, right? So just like the ones that we looked at before, those kind of amino acids / peptides that we try to decipher, right, by looking at the structure to find out what kind of amino acid there is, right? And those happen, this process happens in the cell like 1,000,000 times a second, I would say, right? So there's a lot of this going on. And just to imagine what this process actually looks like, because a ribosome is actually also a protein. I looked for a video which has a 3D representation of what an actual, basically the making of a protein, what it would look like. Right? So, that was not it. Nope. Okay, let's take a look.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay, so a lot of information, but I think it's very, very interesting. And I think it's a very amazing process. So you saw a little bit what you see here as a ribosome is very similar to what we could see in PyMol, right? This kind of representation where you can see what we looked at was what you call a cartoon representation. And this one you would say a sphere representation, right? So you can see the ribosome kind of is basically like a bundle of grapes, right? In this case, one grape is one atom which has a sphere around it. And yeah, here it is shown in blue and green is the tRNA. So you see, like actually also proteins, they're not as static as we would see it in PyMol, right? Because in nature, everything's moving, everything is in action, right? Because of, for instance, temperature and things like that. So what we saw in this video is also that the folding, right? So the from the peptide to the actual protein, this process takes all place at the site where the peptide basically comes out of the ribosome. Right? So you saw many amino acids are basically connected together to form a peptide, and then when it comes out of the ribosome, it forms a certain shape. Right? So let's see. But what the shape actually is going to be is not 100% set. Right? This is the picture that I used for all the streams in the beginning, and I'm not sure how many of you actually understood what I was showing there. What you can see on the left, is basically a peptide. Something that is unstructured and completely unfolded. This would not be a functional protein yet. Right? The goal at the end is, or this is what you would find after the whole folding process is finished, right? This is the final protein. In this case, what scientists or talk about in this case is the native state. Right? So the native state of the protein is the folded and functional, folded and functional protein. But as you can see, to get to this final result, there are many, many ways you can get there, right? Because if you would think about having a string and trying to put it into a certain conformation, you could also start from one end, from the other end, you can start in the middle, you can all basically start randomly and get still to the same result. And today we're going to talk a little bit more in detail how this happens, right? So, and one thing that is very important for you to understand is energy and entropy. I'm not sure how many of you have had these topics in chemistry or even physics yet. I think they're part of both. But basically, well, energy is kind of self-explanatory, right? So the more, well, I guess it's not self-explanatory, but energy can have many forms, right? So, for instance, movement, and also in this case, energy means conformation, right? So basically, we talked about in the previous workshops how in proteins, for instance, in between amino acids, there can be different bonds, right? So these bonds, like for instance, hydrogen bonds, they have a favorable energy, right? So the goal of each protein is kind of to have as little energy as possible. Right? In this case, the unfolded protein has the highest energy, and the native state has the lowest energy. Right? Okay. Have you ever heard something about entropy? Right? Entropy is basically a representation of disorder. Right? This also has a value that is similar to energy. And if you think about, for instance, if you think about this state and this state at the top, the unfolded and the native state, the unfolded state has a higher entropy and the native state has a lower entropy. What that means is basically this has more chaos and this has a more structured form, so it's like less chaos. This is basically what entropy represents. Naturally, though, in the universe, everything tends to want to be as chaotic as possible. That's one of the basic laws of thermodynamics. So, but to make a structure, it's basically a fight between entropy and energy, right? So the low energy, or basically forming this native state, gives out energy, which can then basically counteract the tendency of entropy to make proteins more chaotic. Okay, now that I'm saying it, I probably think it's kind of difficult to get in the beginning. But I think, or I hope at the end of this workshop, you will kind of more understand what this whole process is, right? So as I told you about, actually, when you look at this graph before, right, this is only a two-dimensional graph, where you would say, for instance, you can have an unfolded protein, which would have high energy on the left, and then goes down and basically folds itself together to get to the native state, where it then has low energy and also low entropy. But basically, in this case, it's only two directions. But in reality, similar to what we saw here, there are more than two ways to come to this final state of this protein. So actually, it's more correct to represent this graph in 3D. Right. And something that we also have to add in this graph are some things that happen in nature which are different from the native state, right? Because as a protein folds, there's the native state which is the correct folding, but also something can happen, which is what you would say an incorrect folding, something that's not functional. It has a regular structure, it has a low energy, but it does not represent a functional protein. Right? Both options exist, and they are in an equilibrium in a normal cell. Right? So in this case, the greener part, would be the correct way a protein folds, but then there's also always the option that basically a protein self-associates and then makes these ordered aggregates, which are then not functional anymore. Maybe if you think about it, I mean, I don't know how many of you are cooking, but you know how you turn an egg into basically a scrambled egg or sunny side up egg, right? So something that's basically liquid and kind of slimy before turns to do something that's rather solid and not really slimy anymore. What do you think is happening in this process? Right. So what's actually happening is the protein that's in the egg in its native state, you put in energy by heating it up, so it unfolds at a certain point, right? If you put enough energy into a protein, it starts to lose its structure, returns to the high entropy state, very chaotic. It becomes the single string peptide again. And then it actually, if you heat it long enough, it forms these ordered aggregates. And ordered aggregates, in contrast to, for instance, like single proteins, they form longer chains and have a more rigid structure if you would look at them in a higher amount. So basically, if you're heating an egg and boiling or frying or whatever, you basically turn the native state proteins into ordered aggregates, into non-functional versions of the proteins. So if you think about it, while making a protein, as we saw before in the video, it is very, very important that this protein does not go to the right side of this graph and form aggregates. Right. One very important part of this is, for instance, that it's released slowly as a peptide from the ribosome. Right? So the protein can fold step by step and not all at once. Another thing that helps it is another protein, actually, which exists in the cells. These proteins are called chaperones. Right? And as you can see here, this is a representation of what is happening with the ribosome and the mRNA with the translation of the mRNA. So here in blue, you can see that the peptide is emerging from the ribosome as it goes along the mRNA. And as soon as it comes out, there is a protein that binds to it. This protein that binds to it basically puts it into a specific shape. Right? In this case, it is, I would say, preventing it from associating with itself. Right? So, for instance, what you can imagine, if this peptide comes out, it could just fold onto itself, and it would already have made a structure which is maybe not reversible. Okay, we have a question. Okay, if you heat up and cool down the protein, does it always go to ordered aggregates? Well, it depends on the one part how high you heat it up. And it also depends on, for instance, are there other proteins? And also what protein it is. I would say for the most part, if you cool it down just as it is, it will always lose its structure. Because proteins basically fold into the correct shape under very specific conditions, which are made by the cells. Sometimes they can be assisted by chaperones. Sometimes something exists in cells which is, these kind of proteins are called heat shock proteins. And as their name says, they help cells tolerate basically higher temperatures for a certain amount of time. Their function is limited, but they basically bind to proteins which become unfolded due to high temperatures and make sure that when it cools down again and they leave the protein, that it falls back into the same orientation as before. Right? So there's a whole group and a whole family of proteins that is designed to do this. Different organisms have different amounts and different types of these proteins, but I would say in most organisms they exist. Right? Yes. So, well, how you would cool it down does not really affect it. The most important part is to basically, if you want to prevent something from losing its functional structure, basically you have to protect it while it's heating up. The cooling down part is, well, if it's already heated up too much, there's very little that could be done to bring it back to its old function. Right? So, yeah, you would, I mean, if you imagine a plate of spaghetti, right? If you pick it up one time and twist some parts of it and then put it back onto the plate, there's almost a 0% chance that it's going to go back into the same structure that it was before. I mean, there are some more beneficial factors in proteins that would bring it into a position that is, or into a structure that would be similar to what it was before, but probably not exactly. But the minimum amount of entropy can be visualized by your. maximum entropy would be represented by the untwisted shape. Okay. All right. Yes, good comment, Pavel. So back to the chaperones. So similar to the heat-shock proteins, chaperones don't protect from something, but they help with the folding right after a protein is made. All right, as you can see, the whole peptide emerges, and then the chaperones leave, and then the protein is in its native state, in its functional form. Okay, so actually, if you go to Manai, or in the future, if we will go back to Manai, you can actually take a look at one of these, because I took the time and printed out one of these. This is called GROW-L, GROW-S, which is found in E. coli, actually, in bacteria. This is a chaperone, which can help fold proteins. So what you see here is a 3D printed version of the surface. Right? If you remember the representation from PyMol, which we looked at last time with the inhibitor, right? You could see a little bit that the outside was basically rounded off, right? Instead of like having balls or sticks that represent the atoms, in this case you only have the outside surface of the protein. And this is what it would look like, right? In this case, this chaperone has 3 parts, actually, yeah, which can then fit together to form this larger part. And in theory, what happens is something like this. So this is the same protein, just upside down, right? So this one's open and this one's closed, right? So one part of that I showed before is basically the lid part, and the other one is like a barrel part, and then there's another one. So this, in total, this protein would have basically 4 subunits. The green line represents a protein, right? So this protein can take up another protein by the change of its structure. So in the beginning, it's kind of small and has a small inner volume. And then if it binds ATP, it kind of stretches out and opens up, and then can facilitate the binding of a protein inside. What happens then is that the lid goes on. On the other side, the lid goes off. Then the ATP is turned into ADP, and basically a structural change is happening in the inside of this protein, right? And you could imagine it as something as like a chewing motion. So the volume gets smaller and bigger and smaller and bigger. And what's happening, this kind of chewing through the peptide kind of rearranges the structures in it to form the correct secondary structures that are necessary to have a functional native structure. Right? So in the end, ATP is then again used to get rid of the lid, and another protein enters on the other side, and the lid goes off, and this folded protein goes out. So it's a very, very interesting protein, and a very interesting process, if you think about it, that takes place to ensure that all proteins that are produced get into a functional, or get into the right form to perform their function. Right? In this case, basically going into this inner space being chewed through and then going out. This can happen multiple times on both sides, and in the end you have a functional protein. Very good. So basically, if you want to go back to this graph, you have a bunch of unfolded proteins in the top. And what's happening, maybe here is an energetically what do you say? An energetically supported state, right? So something that a lot of proteins fall into because it has a low energy, but actually the correct native state would be on the other side of this hill, right? So to turn this partially folded state into its native state, you have to put in some energy to basically drive it over this mountain to then fall into the right conformation on the other side. Right. So this is something that, for instance, this protein does, which I showed you before. Or there are also proteins or chaperones that prevent proteins from falling into this aggregated state. Right? So it can be that they do either one of these tasks or they do both. All of these three options are possible. So, very good. So now I would like you to kind of think about this process by actually trying out to make your own little peptide and trying to fold it. Right? So, and what I want you to do for this is go to PyMol. Okay, so in this case, we don't need the file from last time. All I want you to do is open PyMol by itself. So you only have like the typical interface and a black screen. Give you some time to get ready and set everything up. So in this case, I told you PyMol has tons and tons of functions. So far we've only used the viewing functions, like displaying and selecting different chains and rotating and looking at different parts. What we are going to use today is called the builder function. So if you can see my mouse, it's in the top right corner. So there are many small buttons like reset, zoom, orient, draw, unpick, and so on. And one of them says builder. Right? So if you click on this, oh, you actually don't see it, you will get a pop-up window with a bunch of like chemical like elements and chemical formulas and stuff like that. And you can attach it, if you drag it, you can attach it to the window of PyMol. Right? So this is what it would look like. And this helps you, with this tool, you can actually build a protein from zero. Right? Just one that you think of. So what we're going to start with is we're going to need a first amino acid, which I haven't found out yet how to add it from this menu, but you can also add it by going to the taskbar on the top, clicking on build, and then going to residue, and then clicking on alanine. I wonder what, okay, it's strange. You can't see it on the stream, but I actually have a drop-down menu which says residue and then click on alanine. What you should get is, well, an amino acid, alanine, on your screen. If you have any problems and if you don't really know what's happening, please let me know so I can give you a little bit more time to work on it. Right. Otherwise, I just assume that everybody knows what's going on. So next, what we're going to do is we're going to add another amino acid. right? So now in PyMol, you can see on one of the atoms, specifically the C on the right side, there's a ball, right? This ball basically represents where we are going to add our amino acid. So what we can do is, if you go to the top right, there's another bar, not chemical, but you can go to proteins. Right? So you could also add other atoms and bonds and also build a molecule that's actually not a protein. But in this case, we want to build a protein, basically. So at first, we're going to add, for instance, another alanine. In this case, you can see in the top right, you have the 3-letter code for the amino acid. So alanine would be ALA. If you click on that, okay, now we have 2. Okay, so I hope you kind of understand how that works. Okay, and why not add a third one, right? And maybe a fourth one. 6, 7, 8, 9, 10. Okay, so now we have a structure of 10 amino acids. Does it look like a normal peptide? It does not, right? Because this, if you remember from last time, what does it look like? Type in the chat if you know. What does this look like? If you turn around and look at it, what kind of structure does this remind you of from last time? Any ideas? Any takers? What is this? What structure is this? Actually, just so I know how many people are actually working with me. Otherwise I can, go on faster. So if you're working with me in PyMol at the moment, please let me know in chat. Either Zoom chat or YouTube chat, that's fine. Very good. Okay, so the structure is an alpha helix, right? So if you thought about alpha helix, you got it. If not, well, now you know what an alpha helix looks like, right? This right-handed shape that's going upwards. By the way, right now I do not hear anything from Zoom. So the only way to ask me something would be through typing. Oh, okay, Koto, very good. Excellent. Now I saw it. Is the delay so bad today? Shouldn't be, but okay. All right, so here we have an alpha helix. Why did it make an alpha helix? Well, the truth is because we selected it. From the beginning, the computer does not know what kind of structure a peptide would have, but because below all these amino acids, you have an option to give it basically a primary, secondary structure, right? And you have three options like we talked about. You have the alpha helix, the beta sheet antiparallel, and the beta sheet parallel, right? So either one of these you can choose to build a protein. Well, right now we have this twist, right, from the alpha helix. But if you don't want to have that, just use an antiparallel beta sheet. In this case, what I would like you to do is from last time, if you go to the whiteboard, I posted the link on Sokoko. If you look at the whiteboard from last time, we kind of talked about what kind of structures peptides would form if they had hydrophilic, hydrophobic parts, right? Green and red. So in this case, we have right now in PyMol just a chain of 10 alanines, right? Alanine is what type of amino acid? What kind of property does it have? Okay. I would like you to answer in either Zoom or YouTube chat. All you need to do is take a look at the picture on the whiteboard and find alanine. And if you found alanine, I would also like you to tell me one polar amino acid that we want to use in the peptide that we're making in PyMol. Right. So if you have a request, then put it in Zoom chat. Otherwise, you can use any one of the polar amino acids from this list. So again, alanine is what... Oh, in what category of amino acids does it belong to? Okay, very good. Very good. All right. Alanine is non-polar, right? So not to get confused with last time, basically right now in PyMol, what you probably see is green, right? You're building basically a green chain. But in this case, it would be hydrophobic. And to change this, what I want you to do is go to PyMol and you click on this tab on the top here and click on display. Display, and then the first option there just says sequence. And what you will get is like this, basically the amino acid sequence, which it will display here, which is very useful because you can, from this peptide that you're creating, you can just pick out the ones that you want to select by just left clicking and dragging over them. You can select or unselect them. Right, and you will see the pink dots pop up in your PyMol that shows you have selected these amino acids. So now we actually want hydrophobic parts to be red. So right now they're green. So all we have to do is, now that we've selected all the 10 alanines, right? So they're highlighted up here. Go to the right side where it says Seller for selection and click on the C, which will give you a drop-down menu, and you can choose colors. In this case, I'm going to choose reds. Then there's another little pop-up that says different types of reds. I'm just going to get red, right? Okay. So now our non-polar chain is red. Okay. So now let's continue building. We're going to deselect all the amino acids again. And then the request for the polar amino acid from Ashta was cysteine. So cysteine, three letter code is CYS. So in the builder, click on CYS. And we're going to go for 10 as well. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. Very good. So now you have a long chain of cysteines. Okay, they're all red now. So you have to select them again in the sequence on the top and go to selection or seller, click on the C, go to green and select green. Okay, deselect and we can continue. And again, now I would like you to add another non-polar, right? So anyone you want from this list, from the whiteboard, you can choose and just add 10 of them. Right? For instance, I'm going to choose. I'm going to choose glycine. GLY. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. Select all. Selection. Color red. Red. Okay. So you probably see what I'm trying to do. I'm trying to rebuild, if you compare it, right? So I'm trying to rebuild what we drew in the whiteboard. Now I'm trying to build this in PyMol, right? With just random amino acids which have the property of being polar or non-polar. Okay. Right, going back to the whiteboard. Now I'm going to pick some polar side chains again, probably also 10 in a row. I'm just going to go random for now. I'm not going to pick one and do it 10 times, so I'm just going to pick a bunch of them. Okay, let's see. Let's go with more cysteine. Okay, going to PyMol. let's see, cysteine, maybe 3, methionine 2, glutamine, glutamate, histidine. Is that 10? Wait. Okay, that's actually more than 10, but should be fine either way. As long as you select them and make him green again. Okay, again, nonpolar, 10 of them. Valine, alanine, valine, glycine, glycine, alanine, valine. Isoleucine. Okay, maybe like that. Select color red. Okay, and maybe finally, like, let's do 2 more, like 10 polar and 10 non-polar. If we want to use the same amino acids in a row, that's totally fine. Oh. Histidine, tyrosine, glutamine, lysine. More. Okay. So basically, this is what it should look like in the end. So I'm going to give you a little bit of time because that is probably going to take a little bit if you don't know how to use this program or if you're not familiar with it. So in the end, we will have about, well, yeah, 70 amino acids. So in the top, you can see, well, I have, was it 72, I think? Yeah. Okay. If you're done, please let me know in chat so I know when to continue. Ah, okay. Okay, thank you for telling me. Yeah, my mic was muted. All right, so uh, let me just start again. So maybe you saw me hide the sidechains, I hope. If you did not, go to all, click on H for hide, and then you hide licorice. Right? And then it gives you a line like you see on the whiteboard. And now you know the hydrophobic parts are red, the hydrophilic are green. So if you've done this, PyMol actually has a sculpting function where you can kind of put this peptide into a random conformation, or give it a random structure. You get this by clicking on Sculpt in the Builder window. What that will give you is another menu on the bottom right, which says Sculpt, with 5 options: undo, switch object, scramble, unrestrained coordinates. Now you should be able to hear me. Not working. Okay. So we're back. I think, Pavel, you have a higher delay than maybe Ashta. So something that helps sometimes is refreshing the stream. Okay, so now that we have this long protein, we want to give it a structure. So what you can do is you click on, be careful not to click any of the other buttons. Just click on Scramble Unrestrained Coordinates. So what will happen is basically it will like scrunch it together. Like this. Right? And this process is going to be completely random. So whatever you did and how you made your peptide, this is going to look completely different from mine, perhaps. All right, so basically, what I'm showing you here, or what you see on your screen, is now one of the possible conformations a protein or a peptide can have with this amino acid sequence that we determined. What you can do, you can also click on it one more time, and now you have a different one. Right? So first it's a little bit more compact, and then because of the sculpting process that's going on, it kind of moves a little bit more apart, and then you get this. Right? So there's some already, if you, I don't know, it depends on what you see on your screen, but if you followed the last workshops, you can already see some interesting things that we talked about. Right? Perhaps, but only perhaps, you see secondary structures, right? For instance, here, you would see something that could resemble an alpha helix. Only maybe. Doesn't have to be 100%. What you also see, probably, is that the hydrophobic parts are kind of sticking together in the same area. and kind of are aligned with each other. Unfortunately, I'm not really a professional with PyMol. There's probably a lot more information you could get out of this. I would bet there is actually an energy calculator for this state that you've created. And you could switch and look at different conformations, compare their conformational energy and how well the structure basically holds together. For us right now, the only indicator I know of that is kind of easier to do is basically you can look for hydrogen bonds. Hydrogen bonds are a very good indicator on how good the structure is that you're looking at, right? So if you remember, the secondary structures like alpha helix and beta sheets, they always have a lot of hydrogen bonds between them, which makes them very, very stable and very, very common. Right? So I would say basically the more hydrogen bonds you have, the more stable the present conformation of the peptide that you're looking at. So to make those visible, it's actually relatively easy if you go to all and then you go to find, right? So all gives you a drop-down menu and find is one of the options. You have find polar contacts, and then it gives you a bigger drop-down menu which says within selection. All right, so in this case, it will find all the hydrogen bonds within the selected amino acids. Right? In this case, it's all of the amino acids. So if I click this, you will start to see that there are yellow lines popping up in between. So these yellow dotted lines, they indicate this is a place where a hydrogen bond could take place. All right. So in this case, I can just count them. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? So my structure would have 12 hydrogen bonds. I would say that's not so good, perhaps. We can see that most of the hydrogen bonds are also in this structure that would resemble maybe an alpha helix. And the rest is relatively unstructured. All right, so if you want to do this again, you first have to get rid of the hydrogen bonds. To do this, what I always do is I just hide everything and then show and then ribbon. Oh, actually that looks different. Show cartoon. And then we're back to this representation without the lines. So if you want to try another one, just go back to scramble. Let it unfold a little bit. Now it's completely different conformation. You can just go to find polar contacts within selection. You see, actually that looks a lot better than the last one. Right? So basically we are the chaperone. We are the protein that's basically chewing through this peptide sequence, and you can chew it a lot of times, and then you stop at the one that's the best. That has the least amount of energy in this, basically the most stable structured form. All right, so in this case, I got like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 hydrogen bonds. So better. So, just out of curiosity, how many hydrogen bonds did you find? Okay. So this is actually a very interesting tool to play with. And I think PyMol has a lot of options to actually build theoretical proteins. I'm not 100% familiar with all of them. But it's definitely fun to play with. I've played with it quite a bit just to see what kind of structures I can get and how it basically aligns with what I know about how protein structures should look like. And it's also a very good representation of how actual research about proteins is carried out, for instance, by computers. Because a lot of research goes into actually finding the structure of proteins just from their sequence. The reason is, well, proteins, you can basically understand what their function is and how they work by looking at their structure. The protein we looked at last time, it's a very work-intensive and long and basically research that takes a long time to get this structure. In this case, it was X-ray crystallography, right? To be able to look at the structure and analyze the proteins. And if you think about this, that there are thousands and tens of thousands and hundreds of thousands of different proteins, you cannot make a physical or basically X-ray crystallography with all of them. So optimally, what people would just like to do is, if they know the amino acid sequence, for instance from the DNA sequence, just translate it into mRNA and then into protein, they could just feed it into a computer, and then it would basically give them the structure of the protein and what it would look like, right? But since there are millions of different possibilities, that's very hard to do. So most programs that are trying to solve it, they're basically doing the same thing that we are doing in this case. They're basically taking the peptide sequence and they're basically chewing through it and giving it different conformations and then looking at the theoretical energy of this structure. And then if, for instance, there's one part that has a very good energy, they start just, for instance, changing the conformation locally, right? So right next to, for instance, the part that has a very good energy. Something that I would like to show you, because it's a very processor intensive type of analysis, right? So you have to look at the energy, you have to look at so many different conformations, that people actually have thought of an interesting way how to make this more, I would say, cheaper, and also basically involve people that are just interested, not really involved in the research, right? So there's something called, it used to be called Rosetta at Home. Now it's, I think, called BOINC, BOINC. And basically this is a software that you can download, which then uses your processor to determine the 3D structure of proteins, right? So whenever you're not using your computer, for instance, screensaver mode, you can use the processing power it has to help the University of Berkeley, I think it is, solve protein structures and further basically their research. So I wanted to show you how that works. Let me go back to the presentation. So this is what we did right now. We're going to come back to this. This is what it will look like, right? So right now I'm using a lot of my processor. That's why I cannot run the program at the same time. But basically you then have a representation like this. And I hope with what we talked about today, you kind of understand what is going on in this video, right? So what the computer is doing. Let's take a look. All right. So this is a visual representation of what the processor would be doing on a computer. On the left is what is basically we did in PyMol. We put it into different, this peptide, we put it into different conformations. And what you can see on the bottom in this graph is actually the energy, right, of this protein. And then you basically, your computer tries to, like with this graph and this kind of computation, to find the lowest energy possible. And then sends the results back to the lab in Berkeley. Okay, so for instance, now you see there was one conformation which was like crazy good for the energy. So it went down and now it's trying other areas of the protein to fold into something that's similar. Or like similarly, like energetically favorable, right? So you have like a valley here, then it starts chewing again and goes a little bit up because there's not much happening in other parts which make it more energetically favorable. And so on, right? So this is something that you could do if you download it to help research just by having your computer on all day, which you probably have anyways. But definitely, it also takes a lot of processing power. So it's probably not something to run if you want to, for instance, play games at the same time, or maybe even if you want to watch a movie at the same time, because that also takes somewhat processing power. Okay, but it's an interesting option. Something else that I want to introduce to you at the end of today's workshop is something called Folded, because this is more like a game, but it's actual research, right? So somebody thought, okay, actually computers are very nice in theoretically analyzing shapes, but still the human brain is a very powerful computer that is very good at actually analyzing and imagining shapes and conformations that could get to a result faster than just a computer. So I want to show you what that would look like. There's a program that you can download, and it is basically very, very, very similar to PyMol, right? So you have a representation of what this structure would look like, what a protein could look like. And in this case, what you can do is you can just, I hope the sound is not too loud, but you can just take different parts and you can turn them. You can drag them, right? And you can change the conformation. And what you have at the top is basically a score. So it tells you how good or basically the energy of the protein is that you've made. There's also like some, for instance, some tools where it does it a little bit automatically, so you can just pull it in one direction and then you have wiggle itself back into a position that's energetically favorable. That's kind of, you can see at the top, it's creating points by itself. So, and so on. So in this case, this is used to solve real-life problems. For instance, what we're looking at here is actually the coronavirus receptor that recognizes human cells to then invade them and do the whole process of replication. Right? So this is called, like, what's here in gray, this is something you cannot change, which is the spike protein on the outside of the virus, which then attaches to the human cells and recognizes them. What is being built here is basically a peptide that can attach to this receptor and basically block it. Right? So you could help the research against, or like on coronavirus, and to find a cure by playing this game. There's like on the right you can also see there are like high scores, and top list there are also competitions of this game. Right? Who can find the best conformation structures that can then be realized in actual medicine or other research results. Or maybe I can show you, there is also the option, for instance, to also design proteins from scratch. Okay, there's actually one I already like messed with a little bit. Okay. So in this case, just before in PyMol we build the sequence, right, of different amino acids. It just, here it gives you a long one. And what you can do is you can then, in the design mode, you can, for instance, just change the different amino acids into different ones that we talked about, right? For instance, I want this to be an arginine, right? So it changes it. Or I want this one to be a glutamine, and this one histidine. So then, in the pull mode, you can actually try and fold them together to then get points to make a structure, right? If it's not good, it gives you negative points. You can see there are like basically clashes. So areas where structures cannot be in proximity because it's not physically possible, right? And again, you can like start to wiggle and then kind of tries to do it on its own. Okay, and now you have, well, it doesn't look very much like a protein, but if you continue building, you would get to something that could potentially be protein. Also in this structure as it would be in real life, if you were to make it. So I definitely recommend it if you're a little bit bored, or if you want to like look into proteins and how they work and how the structure works a little bit more. Definitely try out this program. I think it's fun, and there's also, if it looks like a lot in the beginning, there's a very good tutorial on what to do and what kind of options you have with the game. And yeah, basically it also makes it into a game, so it's fun. It's not only learning. So very, very good tool. Plus you can feel like a researcher because it's actual research that you can do. All right. So with this, I think we're done for today. If you have any questions, please ask them in chat. Otherwise, I will see you hopefully in Zoom if you have time after this as well. Well, let's see. Let's see if there are any questions. Well, actually, I think maybe better idea is to now stop the stream and then I will come to Zoom and then we can talk there. I mean, I also still see the chat and I can type in YouTube, but I don't have to wait because of the delay and stuff like that. Okay, so in this case, thank you for coming today and I will see you guys